Hello, and welcome back to Cellvita Science Talks, where we explore key challenges in drug discovery and development. In today's episode, we continue our discussion on the topic of cell-based potency assays, this time focusing on a real-life case study, developing cell-based potency assays without losing your sanity. The full version of this article, including all figures and graphics, is available on our website. If you're watching on YouTube, you'll see the visuals on screen. And if you're listening on Spotify, we'll guide you through them step by step. In our previous episode, we explored what defines a cell-based potency assay and why it is considered one of the most complex tools in pharmaceutical development. We discussed how potency, as a critical quality attribute, requires more than standard analytical thinking. Developing a meaningful bioassay demands a deep understanding of the drug's structure, its mechanism of action, and formulation, alongside practical constraints such as laboratory conditions and reagent availability. All of this serves one purpose: ensuring patient safety through biologically meaningful and reliable data. From theory to real projects. Theory provides the framework, but real projects reveal the true complexity of potency bioassay development. Our goal is to translate biological complexity into biologically meaningful, statistically sound, and regulatory-ready potency assays. Close and transparent communication with partners ensures a shared understanding from the start and reduces project risks. Development can vary depending on the therapeutic modality. The two case studies presented here illustrate these differences. Both are classified as potency bioassays, using cell-based models to reflect drug activity in patients. However, beyond this shared foundation, each assay is tailored to the unique characteristics of the molecule and its intended application. These cases highlight a crucial point. The term bioassay alone says very little about methodology. Even similar therapeutic molecules may be assessed using substantially different procedures across laboratories. This variability reflects the need for careful risk assessment and process design adapted to specific environments and conditions. Case Study 1. Primary HUVEC cells for inhibition of proliferation. The challenge. The first case study addressed a demanding question: how to quantify inhibition of endothelial cell proliferation for a fusion protein blocking VEGF signaling in a biologically authentic and regulator-ready manner. Primary HUVEC cells were chosen to capture authentic VEGF receptor biology. Building the assay. Proliferation was maintained using a fixed VEGF concentration. Co-incubation with increasing drug doses produced a dose-dependent reduction in viable cells. Cell viability was measured using CCK8 absorbance at 450 nanometers, ensuring alignment with regulatory expectations and ICH Q6B guidelines. Optimization. The test system consisted of primary HUVEC cells. The mechanism was VEGF-driven proliferation, inhibited by the fusion protein. The readout was absorbance at 450 nanometers. A linear dose-response model was applied using parallel line analysis. Statistical evaluation included parallelism, linearity, regression analysis, F-tests, and relative confidence intervals. Reference standards and assay controls were used throughout. The assay range covered 64 to 156% potency, with an overall accuracy of 96% across 5 potency levels. Specificity was confirmed. Only the target drug inhibited proliferation. The method demonstrated reproducibility and accuracy, making it ready for validation. Data and performance. Dose-dependent inhibition was confirmed using linear regression and statistical testing. Higher drug concentrations led to progressive inhibition, producing a characteristic negative slope in the dose-response curve. The assay generated consistent and reliable results suitable for regulatory use. Figure 1. The figure shows dose-response curves for VEGF-driven cell proliferation measured across two different cell passages. At the top, you can see two curves representing passage 1 and passage 2. In both cases, increasing VEGF concentration leads to a gradual rise in signal, reflecting enhanced cell proliferation. A specific point on each curve marks the selected stimulation level, the infection point at a defined VEGF concentration. This point is used as a reference for further assay development and helps ensure consistency across experiments. Although the overall shape of the curves remains similar between passages, small differences in response levels and EC50 values can be observed. This highlights an important aspect of working with primary cells. Biological variability between passages must be carefully monitored and controlled. Below, additional plots show the relationship between samples across concentrations, with fitted regression lines used for statistical evaluation. These confirm consistent trends and support the use of linear models for reliable potency determination. Together, these visuals demonstrate a clear, dose-dependent biological response and provide a strong foundation for developing a robust potency assay. Why it matters. Using physiologically relevant primary cells ensures that the assay reflects the true therapeutic mechanism. Mechanism-driven design combined with rigorous optimization produces reproducible, regulator-ready data. This translates into faster development timelines, reduced inspection risk, and a clear link between biology and actionable potency results. Case Study 2. Reporter Gene Assay for Immune Signaling. The challenge. The second case study focused on developing a reporter gene potency assay for a novel immuno-oncology peptide. The goal was to translate complex immune activation into a clear and reproducible signal for quantitative potency determination. Unlike the first case, this project began at the most fundamental level: generation and qualification of cellular models. Modified K562 cells acted as antigen-presenting cells, while Jurkat cells were engineered to express a peptide-specific T-cell receptor. Upon exposure to the peptide, activation of the NFAT signaling pathway triggered a luminescent signal. This provided a direct, mechanism-based readout of immune activation. Optimization. The system combined engineered K562 and Jurkat cells. The mechanism was peptide-induced T-cell activation via NFAT signaling. The readout was luminescence. A 4-parameter logistic model was used to describe the dose-response relationship. Statistical evaluation included curve fit quality, EC50, curve symmetry, and parallelism. The assay range covered 60 to 140% potency. Accuracy reached 100% across 5 potency levels. Specificity was confirmed. Only the target peptide triggered the response. The method demonstrated strong reproducibility and was validation-ready. Data and performance. Dose-response data followed a sigmoidal curve, characteristic of biological activation. Key parameters remained consistent across replicates, confirming robustness and reliability. Figure 2. The figure presents several key aspects of assay optimization and performance. At the top, 2 bar charts show how experimental conditions influence the signal. On the left, different ratios between cell types are compared. while on the right, the effect of assay volume on signal intensity is presented. Both plots clearly demonstrate that even small changes in setup can significantly impact the assay readout. In the middle, two dose-response curves are shown side by side. One represents a constrained model, and the other an unconstrained one. Both follow the expected sigmoidal shape, but the comparison highlights how the choice of statistical model can influence how the data are interpreted. In the lower part of the figure, dose-response curves for different sample conditions are compared. You can see clear differences between samples prepared in assay medium and those tested in the presence of formulation matrix. This illustrates how matrix effects can influence the observed potency and why testing under relevant conditions is critical. Finally, a heat map shows signal distribution across the plate. The relatively uniform pattern confirms good assay consistency, while also helping identify any potential spatial effects. Together, these visuals demonstrate how careful optimization, combined with appropriate statistical analysis, leads to a robust and reliable potency assay. Why it matters. Engineering cellular models from the ground up provides full control over biological relevance and assay performance. This approach reduces variability, supports regulatory requirements, and accelerates development timelines. It also provides reliable tools for critical decision-making throughout the product lifecycle. Take-home message. Both case studies demonstrate that successful potency bioassay development starts with biology, not technology. Although the assays differed in design, they shared a common foundation, a deep understanding of the mechanism of action. A potency bioassay is not a method, it is a regulatory purpose. There is no universal template, only solutions tailored to individual products. Robustness is achieved through structured optimization and proactive data quality design. Validation readiness is just as important as validation itself, ensuring smooth transition into routine use. Conclusion. Taken together, these examples reinforce a central message. Potency bioassays succeed when biological relevance, structured optimization, and regulatory thinking are integrated from the very beginning. While complexity is inherent to cell-based assays, uncertainty does not have to be. When designed with purpose and precision, potency bioassays become powerful tools that safeguard product quality and ultimately patient safety. Thank you for listening to Cellvita Science Talks. If you'd like to explore this topic further, we invite you to visit our website and read the full article. And if you're interested in more content like this, stay with us for future episodes, where we continue to explore the science behind modern drug discovery and analytical development.